Hi everyone, so this is Shivdasan. So today I'm going to teach you about one of the most important topic from the lesson biotechnology application. The topic what I'm going to teach you today is RNA interference. As we all know that the chapter biotechnology which has more weightage in the board examination. So today I'm going to talk about RNA interference. Now, what is actually RNA interference? So RNA interference, the term itself indicating RNA interference, something is actually interfering. So before going to this topic, RNA interference, let's discuss about central dogma. So what is actually a central dogma? So as you may have already heard about central dogma, right? So in the chapter molecular basis of inheritance, yes. So here central dogma actually it is discovered by the scientist Francis Crick. So according to Crick, central dogma means, so we all know that actually our characters are stored in DNA in the form of gene, right? So here, see DNA it undergo transcription. You have already studied about transcription, the process of transcription. So DNA undergo transcription and produces RNA, and this RNA undergoes translation, and after the result of translation, protein is synthesized. Protein. So this is actually central dogma. So here, genetic information it passes from DNA to RNA through the process of transcription, and from RNA to protein through the process of translation. Now, in case of viruses, as we know that RNA is the genetic material. Now here, in case of viruses, see the genetic information it passes from RNA to DNA through the process of reverse transcription. Reverse transcription. Now, so what is the importance of explaining central dogma in RNA interferences? See here, as I have already told you that DNA it get that means genetic information it transferred from DNA to RNA and from RNA to protein. Now here, see genetic information means, for example, I'll tell you one example. My eye color or like our skin color. Actually, the character which I got it from my parents, right? So we are getting characters from our parents. Okay, so that character it should express. That means the gene it should express. Okay, now so the characters it is there in our body. Okay, so that character it undergo transcription and produces an RNA, right? So this RNA undergo translation and produces a protein, and after producing the protein only it get expresses. Okay, so here in our cases, as I have already told you, the gene which is responsible for the skin color, okay, it should undergo translation and produces a protein. Now here, in case of this RNA interference mechanism, see here, protein synthesis will not happen. See, the RNA formation will take place, and the RNA it is actually interfered by a component. Okay, that is we can discuss in the upcoming portion. Anyhow, RNA get interfered, and the RNA will not produce a protein, so the gene will not express it. This is actually RNA interference in general. Okay, anyhow, we have to discuss in detail. So this is actually a general idea about RNA interference. Now, actually RNA interference, so this is actually one of the process which can be seen in most of the eukaryotes as a cellular defense mechanisms. Okay. Now this process is also called as actually the co-suppression. We can call it as co-suppression and killing, and then post-transcriptional gene silencing. Okay. Now So this RNA interferences, actually, as I have already told you that it is one of the cellular defense mechanism that can be seen in the eukaryotes. Now, scientists, they have adopted this cellular defense mechanism in order to prevent or in order to kill the pests. Okay, so here this mechanism we can see in most of the pests. That means we can incorporate in pests to produce a pest resistant plant. Okay, so as we know that actually to eradicate the pests we are using lot of chemical pesticides, insecticides and all. Okay, so instead of using this pesticides and insecticides, we know that actually it can cause a lot of problems to environment, right? So instead of using this pesticides and insecticides, we can make a plan which will prevent or which will kill the pests. Okay, so the same method is actually introduced into the plan to kill the pests. So we are moving on to discuss detailing about RNA interferences in a in a plant. Okay. Okay. So as I have already told you that RNA interference or co-suppression or killing or post-transcriptional gene silencing. Now we are moving on to the major processes which happens during RNA interference. Okay. So there is a nematode, we can call it as a kind of roundworm which can infect the root of tobacco plant. Have you know the name of the roundworm? It is actually Meloidigae incognitia. So Meloidigae incognitia, it's a kind of roundworm, as like our scaries and all. It's a kind of roundworm. So this Meloidigae incognitia, which can infect the root of tobacco plant. So what will happen when it will infect the root of tobacco plant? Actually, it will make a nodule in the root of tobacco plant. Okay, bulge. And it can absorb all the nutrients from the root of tobacco plant. So what will happen? Tobacco plant will not get sufficient nutrient. So tobacco plant will not get sufficient water. So as a result, the yield it get decreases. Have you got it? And as well as actually it can cause a secondary infections. Like because of infecting the root, actually it will not allow the absorption of nutrient from the soil. So that is actually secondary infection. So because of this primary and secondary infection, see actually the product or the production it get decreases or the yield it decreases. So that is actually a problem. Okay, so the farmers are facing this problem. So in order to eradicate the meloidic incognition from the tobacco plant, the scientists adopted this methodology like RNA interference. Okay, so just imagine this is actually the root of tobacco plant. Root of tobacco plant. So as I have already told you that the root of tobacco plant it is get affected with the meloidic incognitia. Okay, now actually, see for the RNA interference mechanisms, see a nematode specific gene. Okay, in the nematode, we can separate the gene. That nematode specific gene we have to introduce into the tobacco plant. Okay, so by using a vector, we can introduce the nematode specific gene into the tobacco plant. Just imagine now the gene is there in the tobacco plant. Okay, so we have introduced the gene, the nematode specific gene into the tobacco plant. Now, in the cell of tobacco plant, actually the gene it get activated as I have already told you the central dogma. Okay, so the gene it get activated. How does it get activated? The DNA, that means the genetic information, it will transmit from DNA to RNA, then RNA to protein, right? So from DNA to RNA through the process of transcription and from RNA to protein through the process of translation. So the same process will happen here. Okay, so as a result, what will happen? it will make actually from DNA to RNA, it will produce an RNA. Okay, so usually the DNA is double stranded and RNA is single stranded, right? But here, actually the RNA which is produced in the root of tobacco plant, it become double stranded. How to become double stranded? Either by bidirectional transcription. That means DNA dependent RNA polymerase, it undergo bidirectional transcriptional or loop formation as like that, or loop formation. As a result, it become double stranded. Okay, so just imagine a double stranded RNA, it is actually produced. Just imagine this is the double stranded RNA. Okay, now in the cytoplasm there is an enzyme. That enzyme is called a dicer. Dicer is an enzyme. Okay, so when dicer found the double-stranded RNA, when dicer find the double-stranded RNA, it will cut the double-stranded RNA. That means it will make the double-stranded RNA into lots of small fragments. Okay, so it will make into fragments. So just imagine these are the fragments which is actually produced after the action of dicer enzyme. Okay, now these RNAs, this small fragment of RNAs, we can call it as actually siRNA, then miRNA, and this is shRNA. That means small interfering RNA. Just imagine this is small interfering RNA, and here miRNA, microRNA, and short hairpin RNA. So these are actually the fragments of double-stranded RNA. Okay. Now, see this siRNA actually it has around 20 to 30 nucleotides in between siRNA. Now, see, we can take this siRNA. siRNA is called like a small interfering RNA. So just consider this as the siRNA. Just imagine this as the siRNA. See, keep in your mind all these processes is actually happening inside the root of tobacco plant. Okay, I have already told you. Actually, we are introducing a gene of meloidic incognitia into the root of tobacco plant, right? And in the root of tobacco plant, it will produce a double-stranded RNA either by loop formation or by bidirectional transcription. And then there is an enzyme called a dicer. So the dicer enzyme, it will cut the DNA and it will make many fragments. Okay, so one of the fragment is called siRNA, another one is called miRNA, then it is called a shRNA. Now come to siRNA. Actually, siRNA, it has two strands, right? It is double stranded. Now one of the strand is actually called a passenger strand. Passenger strand. Now the second strand is actually called a guiding strand. So passenger strand and a guiding strand. Okay. Now, so just keep in your mind actually guiding strand. It is actually it is corresponding to the strand which is present in the like meloidic and cognition. Okay, it is corresponding to the strand which is present in the meloidic and cognition. Okay, just keep in your mind. Okay, now two type of strands are there, guiding and passenger strand. Okay, now there is a protein complex. Okay, there is another protein complex. That protein complex is called. Just imagine this is the protein complex. So this protein complex is actually called RISC. So what is actually RISC? RNA induced silencing complex. This is the protein complex. RNA induced silencing complex. Now this protein complex, it has two type of enzyme. Okay, the first type of enzyme is actually called a slicer, and the second one is actually called a argonaute. Okay, slicer and argonaute, the two type of enzyme which is present in the protein complex. Okay, now so this siRNA, it is associated with the RISC. Have you got it? Now the RISC has two enzymes. Now these enzymes it act as an endonucleases. That means it will cut the DNA. That means it will actually cut the passenger strand of the DNA. Okay, it will cut the passenger strand. Now, as a result, only guiding strand will be there in the cells of root of tobacco plant. Have you got it? So these many processes it occurs in the root of tobacco plant. Have you got it? Now, in the root of tobacco plant, only one a strand of RNA is there. That is the guiding strand. Okay, keep in your mind. Now, so just imagine this is actually the nematode. I told you the round verb, right? The nematode meloidic and incognitia. Yes, the nematode meloidic and incognitia. Now, the nematode meloidic and incognitia, it will eat the root of tobacco plant. Okay, it will try to eat the root of tobacco plant. Okay, now all the root of tobacco, that means all the cells in the root of tobacco plant is having what? It has the guiding strand. Now in the root it has guiding strand. Okay, just consider this as the guiding strand. So guiding strand is actually present in the root of tobacco plant in all the cells. Okay. Now, so in the melodic and incognitia, actually it also has DNA, right? So just imagine this is actually the DNA which is present in the melodic and incognitia. The DNA undergo translation. Okay, it undergo transcription, and then it will produce a RNA. Right? It undergo translation and produce a RNA. Are you getting me? So this RNA is there in the body of melodic and incognitia. Now melodic and incognitia, when it will eat the root of tobacco plant, what will happen? The guiding strand which is present in the root of tobacco plant, it will enters into the body of melodic and incognitia. And then what will happen? See, this is actually the guiding strand is complementary to the strand of DNA which is present in the melodic and incognitia. Have you got it? So the guiding strand which is present in the root of tobacco plant, it is complementary to the RNA which is present in the melodic and incognitia. So what will happen? Yes, if it is complementary, it will be associated. Okay, now it become a double stranded RNA. Now, see, we have already studied about risk. Okay, risk, what will happen? Risk, it has an endonucleases, slicer and argonaut. So the risk, what will happen? The risk will digest the RNA. So RNA will produce in the body of melodic and cognitia. Protein will not synthesizes. So the RNA, it is actually digested by risk. Have you got it? That means we can say that the RNA is actually silenced. Okay? Or the RNA is interfered by another RNA. Okay? So this is actually RNA interference mechanisms. Okay? So once again I'll tell you, actually here in the RNA interference mechanism, first we have to keep in your mind that Meloidium incognitia, it is actually the nematode which will infect the root of tobacco plant. Okay, now in the nematode we have to take a gene from the nematode and that we have to introduce into the tobacco plant. Okay, after introducing the gene into the tobacco plant, actually in the tobacco plant it will make a double strand RNA. The double strand RNA, it is actually digested by dicer and it will produce siRNA, miRNA, shRNA. Okay, now this siRNA, small interfering RNA, it is actually has two strands like guiding strand and passenger strand. Now the passenger strand, it is actually digested by the risk. Okay, because it has two type of enzymes like argonaut and slicer. Okay, now see this guiding strand, it is complementary to the RNA which is present in the body of melodic and incognitia. Now, once the melodic and incognitia infect the root of tobacco plant, this guiding strand it get associated with the, or it is actually complementary to the RNA which is present in the melodic incognita and it is associated and which will be digested by risk complex. Okay, so the RNA it is get silenced or it is get interfered by another RNA. So this is actually a mechanism which can be seen in the pest resistant plant. Okay, so thank you. I hope you got an idea about RNA interference. Thank you.